qPCR, quantitative PCR or real-time PCR is an effective technique to quantitatively determine of a target sequence or gene in a sample. Up to date, qPCR is the gold standard method to test patients for viral infections such as COVID-19. For that, a swab is taken which may contain viral material, the RNA is extracted and reverse transcribed into DNA. The DNA can then be analyzed with qPCR. In a conventional PCR, a DNA sequence of interest is amplified. The presence or absence of the DNA sequence is detected in an endpoint analysis after reaction completion. During quantitative PCR or real-time PCR, the accumulation of the amplification product is visualized with fluorescent reporters and the reaction progression is monitored after each cycle. In qPCR, the product amplification is thereof observed in a quantitative perspective. With each PCR cycle, the amount of PCR product doubles, which can be detected as an increase in fluorescence. This generates a classic qPCR curve. A defined signal intensity has to be exceeded until the product is abundant enough so that the fluorescence is detectable over background. The cycle number at which enough amplified product has been accumulated to exceed that threshold is referred as the CT value. qPCR is mainly performed using one of these two assays. In the cybergreen assay, the fluorescent reporter is an intercalating dye that binds to double-stranded DNA. The Tachman qPCR method works with probes specifically designed to bind the sequence of interest upon synthesis of the complementary DNA strand. The probe is cleaved, which results in a fluorescent signal. In both assays, the increase of fluorescence along product amplification generates a curve. During gene expression analysis, multiple genes can be compared the number of cycles after which the signal intensity reaches the threshold is shown as the CT value. For example, the presence of gene 1 is detected after 20 cycles, whereas the presence of the gene 2 is detected in cycle 25. In general, low CT values go along with high DNA concentrations, and high CT values mean that signals are detected late, indicating low DNA concentration. Quantitative PCR finds application in various fields. It can be used for diagnostics to detect infections with pathogens, and it is frequently used for gene expression analysis.